So we are excited. We are looking at a phase 3 study that has a very interesting phase 2 result to look for treatment of chronic late antibody-mediated rejection, which we know is a leading cause of graft failure in the long run. And as of now, there's no treatment for it. What we believe is the antibody-mediated rejection. So there are a lot of study has been ongoing and has been done that looking at how to reduce the effect of antibody by removing antibody itself, use medication to remove the cell that produce antibody, or treat the effect of the antibody through the complement and non-complement mediated pathway. In this particular study, the anti-CD38 is used to address plasma cell and natural killer cell. And the phase 2 study that was published in New England Journal of Medicine showed a very good dramatic result in reducing MVI from 80% in patients who got the drug to 20% to those that did not get the drug. And therefore, we're going to be starting a global phase 3 study of 120 patients. So it's excited. We still have to go to the trial, and I'm hopeful that this may lead to a new effective treatment for our patient that is very much needed. Right. So this is very interesting, right? Because when we talk about the AMR or antibody-mediated rejection, we think the culprit is the antibody. So in order to deplete the antibody, we have been in history trying to remove antibody, use the drug that we know would deplete antibody. And this particular drug is directed against against the molecule called CD38, which basically is on the surface of the plasma cell. So the idea is we kill the plasma cell and we then deplete the antibody. Very good question. So the phase 3 study look at the patient who have antibody-mediated rejection that is chronic, and they include patients who are more than 6 months from the transplant. And the new definition of antibody-mediated rejection include basically two groups. One group is you identify HLA antibody in the blood, and the other group is they may not have antibody that you can detect in the blood, but you have evidence of antibody injury by evidence of C4d positivity in the tissue. This part of the study, looking at patients who has to have both HLA antibody and evidence of the antibody rejection. They cannot have concurrent significant cellular rejection, and they have to have reasonable kidney function. So that's the inclusion criteria of the study. So with that, we are looking at recruiting 120 patients, and the way the study design work is 2-to-1 randomization. So the randomization, 2/3 would get the medication right away. 1/3 would serve as a control group, did not get the medication. They would get medication or placebo for a total of 6 months. At 6 months, they would be rolled over. The group that did not get the medication would get the medication at 6 months. In other words, 2/3 get the medication for 6 months. The 1/3 didn't get it. At 6 months, they will get the medication. They'll be doing 2 biopsies, one at 6 months and one at 1 year. That would be the end of the study to look at the efficacy in terms of the resolution of the antibody-mediated rejection. The other thing that is important to note is they cannot receive any other treatment for antibody-mediated rejection. That is, they cannot receive intravenous immunoglobulin, which we use, or rituximab that we use. I should note that the IVIG or the rituximab is not indicated for treatment of chronic antibody-mediated rejection at this time, and there's no evidence that they would improve chronic antibody-mediated rejection. And the most important thing I would say is at this point, we don't have effective treatment for antibody-mediated rejection.